Welcome to The Explainer, where we turn dense notes into a clear story. Today we're stepping into the world of a very different kind of detective, one that works with a microscope. So let's dive right in.
Think about it like this. A crime has been committed, but the crime scene is microscopic. To solve the case and, treat the patient, we have to become forensic detectives. We're entering the world of CSI Mycology. Our job? To identify the fungal culprit behind an infection. And let me tell you, it's a fascinating process of gathering clues.
So before we can solve any case, we got to know who our suspects are, right? In mycology, identifying these tiny organisms is absolutely critical. I mean, if you get the ID right, you get the treatment right. But if you get it wrong, well, that's when the culprit gets away and the infection just keeps going.
Okay, so our first clue is a classic move for any good fugitive, the disguise. Some of the fungi on our most wanted list are masters of changing how they look to fit their surroundings, and that makes them pretty tricky to pin down.
This shape-shifting is called dimorphism. Out in the cool environment, say around 25 degrees Celsius, these fungi exist as a mold with all these long branching filaments. But as soon as they get inside a nice warm body, 37 degrees, they completely transform into a yeast. It's really the ultimate camouflage, shifting from their environmental form to their parasitic one.
And this gets us down to the nitty-gritty, their basic structure. See, true fungi grow from the very tip, making these smooth, continuous branches we call hyphae. But some of our culprits, like Candida, they take a shortcut. They make these chains of individual cells that are budding off each other, and we call those pseudohyphae. Those little pinches or constrictions you see in the table, that's the dead giveaway. It's not one single continuous organism. It's more like a chain gang of buds that just didn't separate. A really crucial clue for identification.
All right, detective, let's open our first case file. This one's about a group of fungi called dermatophytes. They are responsible for some of the most common infections out there, collectively known as tinea, or, as pretty much everyone else calls it, ringworm.
Now, the first rule of any investigation is to know the location of the crime. And here, where the infection shows up on the body gives it a very specific name. Is it on the scalp? We call that tinea capitis. On the foot? That's tinea pedis, athlete's foot, and so on. Just knowing the location gives us our first big clue about which group of dermatophytes might be our guy.
For scalp infections, we can dig even deeper. We ask, how is the fungus invading the hair? An ectotrix infection means the fungal spores are chilling on the outside of the hair shaft. An endotrix infection means they've gotten inside. And we've got a simple tool to help us out, the Woods lamp, which is just ultraviolet light. Ectotrix infections often fluoresce, literally lighting up the culprits right there on the scene.
And here is where we can get a positive ID. When we grow these fungi in the lab, they produce these reproductive structures called macroconidia, and their shapes are as unique as fingerprints. Seriously. Whether it's the long pencil shape of T. rubrum or that thick-walled spindle of M. canis, this microscopic evidence is often what lets us definitively name our suspect and close the case.
Okay, let's move to our next case. We're graduating from skin-deep infections to a much more dangerous culprit, one that can actually invade the central nervous system and cause meningitis. So yeah, the stakes are much, much higher now.
Our prime suspect in this case is a yeast called Cryptococcus. And what makes it so dangerous, so good at invading the body, is its secret weapon, a thick polysaccharide capsule. You can think of it like an invisible shield, protecting it from our body's own immune defenses.
So, how do you see an invisible shield? Well, we use a classic technique called India ink staining. It's what's known as a negative stain, which means it colors the background, not the organism itself. The ink can't get through the capsule, so what we see under the microscope is this black background with these bright, clear halos around every single yeast cell. Just like that, the shield is revealed.
In any good investigation, you have to rule out other suspects. A common yeast that could be confused with our culprit is Candida albicans. This suspect lineup here shows us exactly how we tell them apart. Does it have a capsule? Nope, only Cryptococcus. Does it pass the germ tube test? That's going to be Candida. A positive urease test? That's Cryptococcus. A few simple tests, and bam, we've got our guy.
So how does a test like the germ tube test actually work? It's a really clever and fast way to ID Candida albicans. Essentially, we trick the yeast. By mixing it with serum and warming it up to body temperature, we're simulating an invasion. And if it really is C. albicans, it'll start to form these little filaments called germ tubes. which is a key step for it to cause an infection. Seeing those tubes is our confirmation.
All right, time for our final case. And this one is a real head-scratcher. It's a chronic, slow-moving infection called mycetoma, and it's famous for being a master imposter. The evidence might seem clear, but the identity of the culprit can be a complete and total surprise.
The scene of a mycetoma infection has three classic signs, what's known as the mycetoma triad. First, you've got a firm, swollen lesion that just grows very, very slowly. Second, you see these things called sinuses, which are kind of like little tunnels that open up to the skin surface. And third, the discharge that comes out of these sinuses contains these visible clumps called granules. And those granules, they hold the key.
So here's the twist. The whole clinical picture looks like a fungal infection, but mycetoma can be caused by two very different types of organisms. True fungi, which we call eumycetoma, or, get this, by certain types of bacteria that just mimic fungi, which we call actinomycetoma. The treatment for each is completely different, so unmasking the imposter is everything.
So how in the world do we tell the true fungus from the bacterial imposter? Well, while there are some clues in how the lesion looks, the real smoking gun is under the microscope. We have to examine those grains from the discharge. Are the filaments thick and wide, over 2 micrometers? That's a true fungus. Are they incredibly thin, less than 2 micrometers? That's the bacterial mimic. That tiny measurement is the critical difference that leads to a completely different treatment plan.
So, after looking at all these cases, what are the essential tools that make all this forensic work possible? Let's take a quick peek inside the mycologist's toolkit that helps us grow, isolate, and ultimately identify these culprits.
You know, the foundation of pretty much any mycology lab is Sabarad's dextrose agar, or SDA. This is the special plate we use to grow our suspects. See, fungi absolutely love sugar, and SDA has a ton of it. It also has a low pH. Most bacteria, on the other hand, hate that combination of high sugar and high acidity, so they just won't grow. This lets our fungal suspects grow out in the open, totally free from contamination.
Throughout all our investigations today, we've seen these key techniques in action. We culture the organism on SDA, we look at its structure with a microscope, we use special stains like India ink to see hidden features, and we run specific biochemical tests like the germ tube test to confirm an identity. Every single tool gives us another piece of the puzzle. So we've seen how mycologists use morphology, staining, and culture to act like true detectives, identifying the culprits behind these infections. But you have to remember, this field is always evolving. New fungal pathogens are emerging all the time, and the old ones, well, they're developing resistance. The detective work, it never really ends. And that just leaves us to wonder, what new mysteries are waiting for us under the microscope?